Welcome to the Biotech Support Group HemoVoid Product Knowledge Series. HemoVoid is hemoglobin depletion reagent for affinity purification of pure hemoglobin for hemoglobin isolation, hemoglobin variant research. HemoVoid has a silica-based protein enrichment matrix. It removes hemoglobin from erythrocyte lysate samples while concentrating low abundance and/or low molecular weight proteins from whole blood, hemolyzed serum, and erythrocyte lysates for proteomic study. HemoVoid protocol uses mild buffers, preserving native enzyme activity of samples in elution fraction. Mild elution maintains tertiary structure and simple transfer to secondary analysis. Currently, researchers are using HemoVoid for hemoglobin enrichment protocol from blood samples for hemoglobin variant research. HemoVoid derives from a silica-based library of individual mixed-mode combinations. The library was designed to facilitate weak binding of proteins, allowing for rapid elution from the matrix without any foreknowledge of the variety of proteins contained in the starting sample. The identification of proteins in erythrocytes for proteomic studies is hampered by a huge abundance of hemoglobin which hides other proteins and makes detection in 2D gel-based separation difficult. It is therefore important to deplete hemoglobin and enrich low abundance proteins. Hemoglobin voids in flow-through greater than 98% with less than a 30-minute bind-wash-elute protocol. Low abundance protein and enzyme enrichment occur upon using HemoVoid. It is disposable, cost-effective, and compatible with high-throughput proteomic applications. Mild elution maintains tertiary structure and simple transfer to secondary analysis. Removes hemoglobin from species including human, sheep, bovine, goat, etc. HemoVoid removes hemoglobin from organs, tissues. The eluted fractions retain their enzymatic and biological activity. The eluted fraction is compatible with LC-MS, activity-based protein profiling, and proteomic studies. This experiment utilizes HemoVoid. When comparing two gel images, the HemoVoid eluate on the right has been severely depleted of hemoglobin. The remainder of the red cell proteins are substantially enriched and are better resolved in the HemoVoid eluate. Many more proteins are detectable after HemoVoid treatment with extensive protein coverage across both membranes. Validated hemoglobin depletion approach for red cell lysate proteome analysis by means of 2D page and Orbitrap MS. It's a published application in the journal Electrophoresis by author Katja Walpurgis. The article states HemoVoid process as a very efficient enrichment of low abundance proteins by simultaneously reducing the hemoglobin concentration of the sample, and that a two-dimensional reference map of the cytosolic red blood cell proteome was generated and a total of 189 different proteins were identified. Thus, the presented approach proved to be highly suitable to prepare reproducible high-resolution two-dimensional protein maps of the RBC cytosol and provides a helpful tool for future studies investigating disease or storage-induced changes of the cytosolic red blood cell proteome. The identification of proteins in erythrocytes for proteomic studies is hampered by a huge amount of hemoglobin, which hides other proteins. Authors Walpurgis et al. Demonstrated HemoVoid's application in the development of a protocol for proteomic analysis of hemoglobin-depleted RBC lysates in human blood from healthy donors. After using HemoVoid, 2D page comparison of the unextracted hemolysate and the HemoVoid depleted hemolysate displays disappearance of the prominent and spared hemoglobin spot formerly containing high amount of hemoglobin on the gel. Thus, HemoVoid allows scientists to detect and study non-hemoglobin proteins subsequent to using LC-MS analysis for the study of cytosolic proteome. Authors recorded that HemoVoid removed more than 98% of cellular hemoglobin, indirectly concentrated minor proteins, and eliminated unbound hemoglobin. Upon comparison of the untreated and HemoVoid-treated RBC lysates in the flow-through and wash fractions by SDS page, scientists discovered the untreated RBC lysate showed two intense hemoglobin-derived bands, approximately 15 and 30 kDa, and the HemoVoid-treated RBC lysate did show several bands which were not visible prior to hemoglobin depletion, possibly representing non-hemoglobin proteins. Flow-through and 3 wash fractions contain some bands plus hemoglobin bands. These are references on HemoVoid. Cooperating G6PD mutations associated with severe neonatal hyperbilirubinemia and cholestasis, published in the journal Pediatric Blood Cancer by authors Mizukawa et al. Functional 20S proteosomes in mature human red blood cells, published in the journal Experimental Biology and Medicine by authors Sudhanilam et al. Order HemoVoid from biotechsupportgroup.com. HemoVoid is available in different quantities: 10 preps, 50 preps, 100 preps. Thank you for listening to the Biotech Support Group HemoVoid Product Knowledge Series.